IB Bio Photosynthesis Part III is the third of 4 movies that cover photosynthesis. Part 3 will focus on the light-dependent reactions. This movie covers higher-level material only. The essential idea is light energy is converted into chemical energy. Photosynthesis uses the energy in sunlight to produce the chemical energy needed for life. Here is the outline of all the movies available for the photosynthesis unit. Only the first 2 movies are mandatory for SL students, and this movie is focused here. Here is the overall photosynthetic equation. Study the equation and get to know the equation well. Remember, the reason that there are 12 water molecules on the left side of the equation is because all of the oxygen evolved in photosynthesis arrives from the photolysis of water. So if 6 molecules of oxygen are evolved by photosynthesis, there must be 12 atoms of oxygen in the 12 molecules of water that serve as a reactant. Remember that white light is composed of many wavelengths, but red and blue wavelengths are important to photosynthesis as they serve to bump electrons to higher levels, levels that are held by electron carriers. This moment, when light bumps electrons to higher levels in chlorophyll, is the singular transformation of solar energy to chemical energy. Chloroplasts have a double outer membrane and internal membranes called thylakoids. Chlorophyll is embedded in the surface of the thylakoid. Leaves appear green because chlorophyll reflects and transmits green wavelengths. Red and blue wavelengths are absorbed. Red and blue wavelengths are absorbed by chlorophyll to bump electrons to higher orbitals held high by electron carriers. The bumped electrons are replaced by the splitting of water. Chlorophyll is the primary photosynthetic pigment. It is embedded in the thylakoid membranes within a chloroplast. Chlorophyll absorbs blue and red wavelengths, resulting in the loss of an electron. Upon the absorption of light energy, chlorophyll loses an electron to an electron carrier as a part of an electron transport system. The electron transport system will produce ATP and NADPH. The electron that is lost from chlorophyll is replaced from water. Water undergoes photolysis. The photolysis of water replaces the electron in chlorophyll and provides hydrogen ions to be used in the synthesis of carbohydrates as a part of photosynthesis. Oxygen is evolved from the photolysis of water. Here are 2 relevant IB syllabus statements. State that oxygen is produced in photosynthesis from the photolysis of water. Outline that photosynthesis consists of light-dependent reactions and light-independent reactions. Photosynthesis broadly has 2 sets of reactions, the light-dependent reactions and the light-independent reactions. The light-dependent reactions require water and light. The light-dependent reactions result in the photolysis of water, releasing oxygen as a metabolic waste product. The absorption of light generates excited electrons that are used to synthesize the chemical energy of ATP and NADPH. NADPH is a molecule like NADH that I introduced to you when we studied respiration. The light-independent reactions take in carbon dioxide along with the energy stored in ATP and NADPH to form energy-containing carbon compounds. The light-independent reactions are the subject of Photosynthesis Movie Part 4. So digging in just a bit deeper, describe the light-dependent reactions, outline the reactants and the products and the energy source for these reactions, describe photolysis. In the light-dependent reactions, light energy, blue and red wavelengths specifically, excite electrons that are moved along electron transport systems to produce the chemical energy of ATP and NADPH. Water is a requirement as it is photolysed to replace the lost electrons from chlorophyll, and oxygen is evolved in the process. In the light-independent reactions, also known as the Calvin cycle, the chemical energy of ATP and NADPH are used to fix carbon dioxide into carbon compounds such as glucose. Hopefully, you understand the relationship between the light-dependent reactions and the light-independent reactions. In this diagram, you can see the inputs and outputs of each, but it's important to remember that the ATP and NADPH from the light-dependent reactions are used to fix carbon dioxide into sugars in the light-independent reactions. It's important to note that the very first carbohydrate formed is a 3-carbon sugar known as triose phosphate, or G3P, glyceraldehyde 3-phosphate. And lastly, the NAD plus and the ADP are cycling back to the light-dependent reactions. In the light-dependent reactions, ADP and PI are used to form ATP, and NADP plus is reduced to NADPH, which are then used again in the light-independent reactions to fix carbon dioxide into sugar. In this nice diagram, you can see the blue and the red wavelengths being absorbed by the chlorophyll on the thylakoid membranes. The light-dependent reactions occur here. Water is an input, and oxygen is evolved by the photolysis of water. The excited electrons move along an electron transport system embedded in the thylakoid membrane, resulting in ATP and NADPH production. ATP and NADPH are used in the light-independent reactions, also known as the Calvin cycle, that occur in the stroma of the chloroplast. The ATP is used for energy, and the NADPH is used as a reducing agent. Carbon dioxide, an input into the light-independent reaction, is fixed into carbohydrates, carbon-containing compounds with chemical energy. The ADP and NADP plus cycle back to the light-dependent reactions. This is a nice slide to study. Light arrives to the thylakoid membrane. Water is split by the light, replacing the lost electrons from chlorophyll. Oxygen is evolved. NADP plus is reduced to NADPH in the light-dependent reactions. ADP is phosphorylated in the light-dependent reactions. The energy and reducing power of ATP and NADPH are used to fix carbon in the light-independent reactions, also known as the Calvin cycle. In the light-independent reactions, ATP is hydrolyzed, providing energy, and the NADPH is oxidized, and the hydrogen from the NADPH is donated to the carbon dioxide to form the carbohydrate. So let's dig in a bit further, explain the light-dependent reactions in detail, include the photoactivation of photosystem II, photolysis of water, electron transport, photoactivation of photosystem I, and the reduction of NADP+. On the thylakoid membrane are two photosystems, photosystem II, photosystem I, and electron transport chains. The photosystems are collections of chlorophyll molecules with the role of absorbing light and concentrating the energy of absorbed light so that electrons can be moved to electron transport systems. Let's zoom in. Embedded in the thylakoid membrane are two photosystems, both of which absorb light to excite electrons. We can see the absorption of light here and here, exciting electrons to higher levels. By the way, photosystem II was discovered after photosystem I, thus PS2 is on the left with PS1 on the right. Once again, photosystem II, photosystem I, with an electron transport system that connects them. The electron lost from photosystem I is replaced by an electron arriving from photosystem II. The electron lost from photosystem 2 is replaced by the photolysis of water. The electrons excited in photosystem 2 are moved to the electron transport chain. Carriers move the electrons downhill to produce a proton gradient that ultimately forms ATP. If you recall ATP production in the mitochondrion, you understand how ATP is produced here. More on this to come. The electron from photosystem 2 replaces the lost electron from photosystem 1 that's been boosted to high orbitals by light. A second electron transport chain donates the electron to NADP+ along with hydrogen ions, reducing NADP+ to NADPH. Remember, all of this occurs in the thylakoid membrane. This slide displays the two photosystems, 2 and 1, with the electron transport system connecting them. A small electron transport chain exists here to reduce NADP+ to NADPH. This diagram is often called the Z scheme. You can see why, and it's an important diagram to study. Light is gathered at photosystem 2, exciting electrons to higher orbitals, given over to electron carriers in the electron transport system. The electrons lost from photosystem 2 are replaced by the photolysis of water. Water is the ultimate electron donator in photosynthesis. The electrons moving through the carriers of the electron transport system produce a proton gradient that results in ATP production using ATPases. Do you recall chemiosmosis from your study of the mitochondrion and cell respiration? Same thing here. I'll strengthen this in just a few slides. The electron from the ETS is donated to photosystem 1 that has lost an electron due to the absorption of light. The electron excited by photosystem 1 serves to reduce NADP+ to NADPH. Remember, all of this happens in the thylakoid membrane, but the photolysis of water, seen here, occurs within the lumen of the thylakoid. You can see the proton gradient beginning to form already as the hydrogen ions from the photolysis of water contribute to the accumulation of protons within the thylakoid lumen. Here are the thylakoid membranes within a chloroplast. We can see a lumen here and here and here. These are compartments within those thylakoid membranes. Photosystems 2 and 1 are embedded in the thylakoid membrane, as are electron transport systems. And the electron transport systems pump hydrogen ions into the lumen, resulting in the accumulation of hydrogen ions within the lumen of the thylakoids. The photolysis of water also occurs in the lumen of the thylakoids. The accumulation of hydrogen ions within the lumen results in ATP production using the ATP synthases seen here. In this detailed image of the thylakoid membrane, you can see photosystem 2 and photosystem 1 both absorbing light. You can see the electron transport system that connects both photosystems. Electrons excited in photosystem 2 are moved through the electron carriers, the ETS, causing hydrogen ions to be pumped into the lumen of the thylakoid. This creates a proton gradient that generates ATP using an ATP synthase. Water is photolysed in the lumen of the thylakoid to provide electrons to photosystem 2. Oxygen is evolved from the chloroplast, and the hydrogen ions resulting from the splitting of water right here accumulate in the lumen, adding to the proton gradient within the lumen. Electrons excited at photosystem 1 serve to reduce NADP+ to NADPH. ATP and NADPH move to the Calvin cycle, the light-independent reactions, in order to fix carbon dioxide into carbohydrate. Here is my drawing of the thylakoid membrane. It displays the absorption of light by photosystem II and photosystem I. Electrons are moved from photosystem II to the electron transport chain and are replaced by the photolysis of water. Electrons in the electron transport chain result in the pumping of hydrogen ions, protons, into the lumen of the thylakoid. Protons accumulating in the lumen diffuse through an ATP synthase for ATP synthesis. NADP plus is reduced to NADPH by electrons from photosystem I. Electrons lost from photosystem I are replaced by electrons arriving from the ETS and photosystem II. NADPH and ATP are important molecules in the Calvin cycle, where they serve to fix carbon dioxide into sugar. In this diagram, you can see the accumulation of protons, hydrogen ions, in the lumen of the thylakoid discs. The proton gradient drives the diffusion of protons through ATP synthases, seen here, to produce ATP. The proton gradient drives the diffusion of protons through the ATP synthases to produce ATP. This is chemiosmotic phosphorylation, just as you learned it when we studied respiration. Some refer to this as photophosphorylation because the energy used to create the proton gradient ultimately came from sunlight. Again, back to the Z-scheme diagram. As you use this diagram to study the light-dependent reactions, you must be careful with the low level of detail here. The ATP production, indicated so simply here, hides the high level of detail in the pumping of protons into the lumen of the thylakoid and the chemiosmotic phosphorylation. Study this diagram carefully so that you can explain light-dependent reactions in detail, including the photoactivation of photosystem II, the photolysis of water, electron transport, photoactivation of photosystem I, and the reduction of NADP+. And in pulling way back to the level of the entire chloroplast, hopefully you now understand the light-dependent reactions, the role of chlorophyll, the role of light, and the role of water as inputs to the reactions. I covered these two IB syllabus statements in Part 2, but here they are again because they are for students studying biology at the higher level. Draw and label the structure of a chloroplast to indicate the adaptations of a chloroplast to its functions. Explain how the structure of a chloroplast is adapted to its function in photosynthesis. Having studied chemiosmosis in cell respiration, along with the mitochondrion, and with Part 2 earlier presented, and the examination of the light-dependent reactions in this movie, these statements should feel comfortable. Keep in mind that the light-independent reactions in photosynthesis occur in the stroma of the chloroplast. Here is a micrograph of a chloroplast within a cell. You can see all the thylakoid membrane within the chloroplast. The large quantity of thylakoid membrane serves to increase the surface area for light absorption by the photosystems and the surface area for electron transport systems and ATP synthases. This micrograph does not make clear the lumen of the thylakoid discs, but keep in mind the role of the lumen in establishing the proton gradient. A compartment is critical for concentrating, accumulating hydrogen ions such that you have a gradient driving the diffusion of hydrogen ions through the ATP synthases. The stroma, the fluid surrounding the thylakoids, has the enzymes involved in the light-independent reactions. In this micrograph of a chloroplast, you can see the stacks of thylakoids well, and these stacks are called grana. You can see the thylakoid lumen well. The lumen of the thylakoid is very important to establishing the proton gradient. The thylakoid membranes increase the surface area for the photosystems 2 and 1, as well as electron transport systems and ATP synthesis. You can see the starch granule here. It's an energy-rich, carbon-containing product of photosynthesis. Here is a nice diagram to study showing the structure of the chloroplast with some information about function. Turn the movie off now to review this slide. If you are considering how to draw a chloroplast, here is an image to study. Or maybe this image would be helpful to study. Or maybe this image would be helpful to study. At this point, you should have a strong understanding of the structure of the chloroplast, particularly its role in the absorption of light by the thylakoid membranes and the nature of the light-dependent reactions. As you think ahead to the light-independent reactions, reactions that occur in the stroma of the chloroplast, you can see from this slide that there are still some important details out in front of us. Part 4 will examine the Calvin cycle in detail, but some aspects of this diagram should already feel familiar. Do you see the inputs of ATP and NADPH, providing energy and reducing power respectively to drive the fixing of carbon dioxide into carbohydrate? And that brings us to the end of IB Bio Photosynthesis Part 3.